Bismillah Rahman Rahim. June 24, paper 4-1, video 1. Starting with question number 1, oxygen is needed for aerobic respiration in cells. In eukaryotic cells, the mitochondrion is the organelle used for aerobic respiration. Figure 1.1 shows a transmission electron micrograph of a mitochondrion. Now I've labeled it A is outer membrane, B is of course outside the mitochondria, so it must be the cytoplasm. Then C is the inner membrane, and D is the matrix, which is the inside, the sort of the gel-like thing inside. Now it says table 1 point lists the 4 stages of aerobic respiration. Now glycolysis, as we know, with reference to figure 1.1, table 1.1, using the letters A to D show where each stage occurs. Now we all know that glycolysis occurs in the cytoplasm, so that will be B. And the link reaction and the Krebs cycle occur inside the matrix, so that will be D. And oxidative phosphorylation will be C, because that's on the inner membrane of the mitochondria. So 3 marks, so that is a difficult, that means it's a very easy level. So 3 marks if all 4 letters are correct, 2 marks if 3 letters are correct, and 1 mark if 1 or 2 letters are correct. So this instruction is given to us by Cambridge as to how to mark this question. So everybody must know that, how we mark this question. Now coming to the B part of the question, in anaerobic conditions, no ATP can be synthesized by oxidative phosphorylation because the process stops. Explain why ATP synthesis by oxidative phosphorylation stops in anaerobic conditions. Now it's very interesting. The reason why it stops is oxygen is the final electron acceptor. Electron transport chain stops, or you can say ETC stops. That's the standard abbreviation. ETC stops. So no hydrogen ions move into the intermembrane space. No electrochemical, no proton gradient, or you can say electrochemical gradient formed. So no protons will move through the ATP synthase. So no regeneration of NAD or FAD will take place. Now, if you know electron transport chain, only then you can do this question. I mean, you need to then revise the chapter on respiration if you are not clear what I'm saying. So it's just the opposite of if I asked you a direct question, explain the process of oxidative phosphorylation. Here we'll have to say that is not going to take place. In oxidative phosphorylation, the oxygen is a final electron acceptor, and the electron transport chain, what happens, the electrons are transported in the carriers across the inner membrane, and NADH and FADH give up their hydrogen, which is broken down to proton and electron, and the protons are pumped into the intermembrane space, and that produces a proton gradient. And then of course the H ions cannot go out because the outer membrane is a phospholipid bilayer. The inner membrane doesn't allow them back into the matrix, so the only channel through which they can come is the ATP synthase, which is actually a hydrogen ion channel, but the word A is synthase, so there is an enzyme at the base of it which catalyzes the reaction of ADP plus inorganic phosphate, and ATP is formed. And the reason why this is called oxidative phosphorylation is because oxygen is needed for this process. In glycolysis, ATP is made, but that is why called substrate level phosphorylation, because oxygen is not needed for that. So let's see how we worded it. Oxygen final electron acceptor, ETC stops, no protons move into the intermembrane space, no proton gradient, no protons move through ATP synthase, and no regeneration of NAD/FAD. Part C, up to 60% of the ATP that is produced in cancer cells comes from lactate fermentation of glucose, even though oxygen is present. Scientists are developing cancer treatments to inhibit the enzyme that catalyzes the last step of lactate fermentation. Explain how the inhibition of an enzyme reduces the production of ATP in cancer cells. Now, a lot of information is given in the question, and you need to figure this out. Now, that enzyme is lactate dehydrogenase. So, no pyruvate is converted to lactate, no NAD regenerated, no pyruvate, so there is going to be no triose phosphate to pyruvate, no ATP from substrate level phosphorylation in glycolysis. Now, every point has to be understood. The enzyme lactate dehydrogenase is going to be, they say they're going to have an inhibitor. So no pyruvate to lactate, no NAD regenerated, no TP to pyruvate. Now this is the actually glycolysis cannot take place because in glycolysis NAD is needed, but NAD is not going to be regenerated. So no ATP from substrate level phosphorylation. Question number 2, coordination in humans involves two main systems, the nervous system and the endocrine system. Paracrine cell signaling is a third way in which coordination occurs. In paracrine signaling, one cell secretes a chemical. So one cell secretes a chemical that diffuses a short distance to act upon cells that are very near to the secreting cell. So from here, it goes to the cells very near it. Identify one similarity and one difference between paracrine cell signaling and cell signaling that occurs as part of the endocrine system. Now, similarities both involve a signaling chemical or a ligand or a signaling molecule. The difference is endocrine chemicals travel in the blood, but paracrine chemicals travel by just diffusion from one cell to the cell very near it. Endocrine, like insulin and glucagon, they travel in the blood or in the circulatory system, and they will be produced, say, in the pancreas, and then they will be carried by blood to the liver cells where they will work on the liver cells. But here they are saying they're just nearby, so there will be no question of blood, it will be just through the tissue fluid. Then it says, explain why neurotransmitters such as acetylcholine could be described as a paracrine signaling molecule. Yeah, why? Because acetylcholine travels a short distance by diffusion across the synaptic cleft, or across the neuromuscular junction. So they've asked you for, in the first question, they've asked you the similarity and difference, and then they've asked you why would it be called. I mean, if you know how the synapse works is the presynaptic neuron and the postsynaptic neuron and the vesicles in it. And the vesicles by exocytosis release the chemical, and that chemical diffuses across this gap, this synaptic gap. So it is very adjacent to it, it's very near it, the presynaptic neuron and the postsynaptic neuron. There's only a microscopic gap between them. So let's look at the wordings now. Similarity both involve a ligand, or you can say a chemical, you can even say a cell signaling molecule. Endocrine chemicals travel in the blood, paracrine chemicals travel by diffusion because they're only moving from a cell to another cell very near it. Then in part 2, acetylcholine, we can write ACH, it's a standard abbreviation. Acetylcholine or ACH travels a short distance by diffusion across the synaptic cleft. So that was 2 marks for that, 2 marks for that. Now the B part of the question, I mean, when I look at such a big thing, this gives me a lot of anxiety attacks. And reading all this is going to give me further anxiety. But please try to make it a habit. Don't get anxious, but try to make it a habit to read it about 2 times and then underline the keywords and then go back to the question and then read the question and then come back to this. Human muscles show paracrine signaling. After muscle cells have been exposed to a substance called palmitate, they produce a signaling molecule, show an increase in the expression of genes involved in a stress response, which is called the stress gene. Scientists carried out 3 experiments to investigate the signaling molecule produced by muscle cells after they've been exposed to palmitate. Experiment A. Now, this is nothing to do what you studied in the A2 syllabus, but it is a new situation and you need to really understand this new situation and then understand the question which is going to be asked. Experiment A, muscle cells were exposed to palmitate. The palmitate-exposed muscle cells were removed from the medium containing palmitate and then cultured in a nutrient medium for 6 days. The palmitate-exposed cells were removed from the nutrient medium and new muscle cells were placed in the used nutrient medium. The expression of stress genes in the new muscle cells was measured after 24 hours. A control using cells that had not been exposed to palmitate was also carried out. Experiment B. Experiment A was repeated, but the used nutrient medium for the palmitate exposed cells and the control was boiled and then cooled before adding the new muscle cells. Experiment C. Experiment A was repeated, but they used nutrient medium from the palmitate exposed cells, and the control was treated to separate the lipid part. This lipid part was added to a new nutrient medium to culture the new muscle cells. The results are shown in figure 2.1. Mean stress gene expression in muscle cells in arbitrary units. Experiment A, experiment B, and experiment C. Medium from cells exposed to palmitate and control medium from cells not exposed to palmitate. With reference to figure 2.1, outline the conclusions that can be drawn about the cell signaling molecule involved in human muscle cell paracrine signal in response to palmitate. Now in the question, when they ask you for conclusions, you've got to be very carefully looking at this graph. Palmitate exposed cells secrete molecules or chemical or substance. You can see here these were all producing 1.5, 2 point something, and 1.5. So all the medium from cells exposed to palmitate was producing more of was expression of the mean gene was more. Now, so this molecule switches on the stress gene in the new muscle cells. Molecule is not denatured by boiling. In B, it was boiled and cooled, but still it's showing the effect. So molecule is not destroyed or denatured by boiling or high temperature. So molecule is not a protein. I'm sure everybody knows that, but they won't write this in the paper. So molecule is not a protein. Molecule is lipid soluble or hydrophobic or non-polar. So let's look at the wordings now. Palmitate exposed cells release chemical. This molecule switches on stress genes in the new muscle cells. Molecule not denatured by boiling, so molecule not a protein, molecule is a lipid. Now coming to part C, the process of protein translation is inhibited in developing muscle cells when stress genes are expressed. Suggest how developing muscle cells that express stress genes will differ in structure from the normal muscle cells. So then you have to come up with all the proteins which are present in muscle cells. So there'll be less actin, there'll be less myosin, there'll be less troponin, less tropomyosin, smaller sarcomeres. So you could have mentioned any of these things. There are 3 marks for this part of the question. So wording it because it's saying process of protein translation is inhibited in developing muscles. So less actin, less myosin, less troponin, less tropomyosin, and smaller sarcomeres. Or you could have even said fewer mitochondria and fewer ribosomes. Anything which is made up of proteins, any organelle which is made up of proteins. Now coming to question number 3, stomata and leaves respond to changes in environmental conditions by opening and closing. This regulates carbon dioxide uptake and water loss. Describe the mechanism by which stomata open in sunlight. It's a very direct question. There's no ambiguity about it. It's a very direct question. So protons pumped from guard cell into the cell wall. So a negative potential inside the guard cell, or you can say chloride ions enter the cell. So voltage gated potassium channels open, potassium ions enter the guard cell, water enters the cell by osmosis because as potassium ions enter, this is going to lower the water potential. More ions inside means lowering the water potential. So water enters the cell by osmosis, guard cells become turgid and increase in volume, and that results in the stomata opening. So H positive ions pumped out of guard cell into the cell wall. Negative potential inside the cell, potassium channels open, water enters the cell by osmosis. Why have I underlined this? Is the reason why I've underlined this because you forget to write this. And without this, you probably won't get a mark. Guard cells become turgid, or you can say increase in volume, or you can say the word swell. But you can't use any other word. You can't say they become fatter. That's pathetic if you say they're fatter. B part of the question, stomatal conductance is a measure of the rate of water vapor loss from the intercellular air spaces of leaves to the atmosphere through the stomata. A student investigated the effect of different concentrations of abscisic acid on stomatal conductance in Helianthus anus, a common sunflower. The student treated the roots of 15 sunflower plants with different concentrations of ABA solution. The plants were left in standard conditions for 24 hours. After 24 hours, the student measured the concentration of ABA in the xylem sap of each plant and the stomatal conductance of the leaves of each plant. So stomatal conductance was what? Is a measure of the rate of water vapor loss from the intercellular air spaces to the atmosphere. The results are shown in figure 3.1. Now be very careful, look at the graph. Concentration of ABA in the xylem sap micromoles meter cube and stomatal conductance in mole meter squares per second. Now you see what is happening. As the concentration of ABA in the xylem sap is more, the stomatal conductance was the maximum when it was less, and it was the least when it's this, of course, more in the xylem sap. So stomatal conductance is more when ABA is less. When ABA is more, stomatal conductance is less. So it's sort of the opposite. Now part 1 says with reference to figure 3.1, describe. If you looked at any or more of other of my videos on paper 4 or paper 2, describe is no biology. Describe means there is no biology. You need not be a biology student to really do this describe. So describe the relationship between the concentration of AB and the xylem sap and stomatal conductance. So you need to go back to the graph. As abscisic acid concentration increases, stomatal conductance decreases. There's no bio in this. So steep decrease in conductance to 100 to 150 ABA and no effect on conductance above 250 to 425. So little or no effect on conductance above 250 you could say, or above you could have said 425. So I mean there's no bio in this. And then you can have to give me, if I say a data code, it means two sets of X and Y figures to support the marking point 1, which is as abscisic acid concentration increases, stomatal conductance decreases. So you give me the figures for this point, or say you can give me here at 125, and then you can give me the figures at 750. So you'll have to give me the units for both of them, units here, and the figures for it. So at you can give me at 125 and at 750. So at 125 micromoles per meter cube ABA, this was the stomatal conductance, and you'll have to read this off this point here. So if you had to give me a data code, you would have given me this and this. And of course, you would head it, the stomatal conductance and the concentration of AB in the xylem. I've just given an example for you all to understand that you would have to give me two values. So you can do any which you like. I mean, of course, I wanted it at 125, and of course, this is the one which I found was easier to do because very easy to figure this out. It was here. So this is how I would do it. Just any two figures from the x-axis and the appropriate thing on the y-axis. So remember, describe is no biology. Don't give me any explanations. Just give me what you're seeing in the graph and just give me a data code to take 2 points on the x-axis and the corresponding points on the y-axis. Then as a suggest is a very difficult question. It's never easy. It's only for differentiating between the A and the A star students. So usually students will not be able to do it if they don't know biology very well. Suggest is always difficult. I always say leave the suggest. Just do the rest of the paper and then come back to the suggest. So suggest explanation for the results. Now ABA causes stomata to close. ABA binds to receptor on guard cells. Calcium ions enter the guard cells, acts as a second messenger. Potassium ions leave the guard cell. Water leaves, so cell becomes flaccid, or you can say less turgid. Then coming to the last part, some transcription factors increase the rate of transcription of genes involved in the closure of stomata. state where transcription factors bind to cause an increase in the rate of transcription. Of course, it will bind to the DNA, to an area just before the gene where transcription factors bind and result in the increase in the rate of transcription. Now, this was not such a difficult suggest. I feel it was a very direct suggest. ABA causes stomata to close. ABA binds to receptor on guard cells. Calcium ions enter the guard cells, which act as a second messenger. Potassium ions leave the guard cells, and water leaves, so cells become flaccid. And here you could have written DNA or you could have written promoter as well.